This is a presentation called The Use of Recombinant Proteins in Medicine by Jacqueline Connaughton and Belinda Seeto. So firstly, I will provide an overview of what we will discuss in today's presentation. So first, I will introduce recombinant proteins and look at the purpose of the technique in general and in medicine. Next, we will review the history and development of the technique, how it works, its current applications, followed by advantages and shortcomings. Finally, we will look at the potential future applications and end with a summary and the works cited. So what are proteins themselves? Well, proteins negotiate almost every action that is carried out in a cell and are capable of an enormous variety of functions, from maintenance and repair to immune function. They also govern the transportation of hemoglobin and regulate metabolism also. They are linear polymers that are made up of 20 amino acids and are in fact the most abundant of all biological molecules. Protein encompasses 10 to 30% of cell mass and form the key structural elements in cells, participating in all cellular activities. They are also the mechanisms through which genes are expressed. And this brings us to what are recombinant proteins? Well, recombinant proteins are genetically engineered proteins that are expressed in a vector by recombinant DNA via a cloned gene. They are revolutionary in the field of biochemistry and biotechnology, and they include examples such as insulin, human growth factor, and recombinant factor VIII. And Belinda will look at these a little bit further on in the presentation. Recombinant DNA technology is the process that creates recombinant DNA molecules and thus the expression of protein by introducing the segments of DNA into a vector for replication. So recombinant DNA is otherwise known as rDNA or in vitro recombination, and it's a molecule of DNA that has been artificially created containing DNA originating from two or more sources. For this reason, it is often referred to as chimeric DNA, and recombinant proteins are expressed and produced as a result of this recombination. So let's briefly look at the purpose of recombinant DNA technology in general. So this technology has many applications. We see it in the food industry, in the creation of enzymes and genetically modified foods for extended shelf life. Also in energy applications where the production of bioethanol and biohydrogen are used as fuel substitutes. We also see it in agriculture where crops are genetically engineered with increased resistance to herbicides. We also see it in research with constant advancement and development of new products in the recombinant technology field. Biotechnology is also an area where this is used in the creation of chemicals and detergents. And of course, in medicine and health, from the creation of vaccines to the development of gene therapy, and this we will expand on further throughout the presentation. The purpose of recombinant proteins in medicine include vaccines and hormones, pharmaceuticals, diagnostic tools, therapeutic applications, gene therapy, and the production of antibodies and so forth. Belinda will look at aspects of these in more detail a bit later. So now we'll look at the history and development of the technique. There were many scientists researching aspects of DNA long before 1969. However, it was really in the 70s that recombinant technology started to take place. Paul Berg and Peter Laban formulated ideas separately for creating rDNAs in vitro and utilizing them for cloning, propagating, and gene expression transversely among species. And that was in 1969 to 1970. From 71, Douglas Berg and colleagues isolate the first plasmid bacterial cloning vector, and David Jackson and colleagues established the first chimeric DNA in vitro. In the same year, Robert Pollack highlighted concerns he had regarding the potential biohazards of cloning. In 1972, Janet Mertz, a student of Paul Berg's, and Ronald Davis identify a new concept in creating chimeric DNAs in vitro called SV40. Stanley Cohen in 1973 and colleagues isolate a new cloning vector, PSC101, and create bacterial intra- and interspecies of rDNAs. In 1973, John Morrow and colleagues clone and propagate ribosomal DNA genes from a eukaryote in E. coli. Then in 1975, Cohen and Boyer filed patent applications relating to recombinant DNA from Stanford Uni and the Uni of California, with themselves as the inventors. Boyer and Swanson established Genentech, the first biotechnology company, in 1976, and in 1980 they had their patent issued by the US Patent Office. It was a mere 3 years later that insulin was produced for the very first time, which was an enormous feat for medicine in managing diabetes. I will now hand you over to Belinda, who will talk about how the technique works.
Recombinant proteins are produced as a result of recombinant DNA expression. A DNA enhancer, which is a special DNA segment from a viral or bacterial source, is added to the plasmid DNA ring to increase gene replication and protein production. The bacterial plasmid vector is cloned and large quantities of the plasmid DNA are produced then placed into host cells, which produce the foreign protein from the recombinant DNA. To allow large production, the host cells are placed in a bioreactor or fermenter to grow, and the recombinant protein is then isolated. These proteins are then applied in medicine to produce revolutionary treatments, drugs and therapies for illness, disease, and cancer. These applications will be discussed in the next few slides. In this slide, I will explain how recombinant DNA technology works and how from this we can produce recombinant proteins. Recombinant DNA involves the DNA from two organisms used to create a new combination. This process begins with gene isolation, where the gene of interest is isolated. This gene, for example, may provide instructions for producing hormones such as insulin. The gene must then be inserted into a bacterial plasmid or phage, which are circular rings of bacterial DNA that serve as vectors. The most commonly used plasmid comes from the bacteria E. coli. But before the gene can be inserted into this plasmid, the bacterial plasmid or phage must be cut to allow insertion of the gene. This is achieved using a restriction enzyme or endonuclease, which act as scissors and deactivate the bacterial plasmid DNA. These enzymes allow cuts at specific DNA sequences, making a single cut in the plasmid, leaving a pair of identical single-stranded ends, allowing hydrogen bonding to a complementary sequence. Once the plasmid is cut, the gene must be inserted. The enzyme DNA ligase incorporates the human gene to the bacterial plasmid. In the next 3 slides, I will briefly discuss a few examples of important recombinant protein products, which were mentioned earlier by Jacqueline. These include recombinant insulin for diabetes, recombinant growth hormone for treating growth hormone deficiencies, and recombinant factor VIII. Applications in recombinant protein in today's modern world are present in overcoming issues in agriculture, the environment, energy, health, and medicine. As Jacqueline went through other industry applications briefly earlier, I will focus on applications in medicine and health. Recombinant DNA technology has helped in treating diseases and improving health. These applications seen particularly in hematology, oncology, endocrinology, immunology, and infectious disease. It is used in diagnosis, including gene therapy, and also monitoring devices and therapeutic strategies. Gene therapy is seen to improve inheritable diseases where the protein is dysfunctional or not produced because of an abnormal gene. For example, diseases including cystic fibrosis, hemophilia, sickle cell anemia, and diabetes. Cancer treatment has been improved through gene therapy, with different strategies including the insertion of tumor suppressor genes to immunotherapy, oncolytic virotherapy, and gene-directed enzyme prodrug therapy. Other cancer gene therapy strategies are vaccination with recombinant viral vectors encoding tumor antigens, and vaccination with host cells produced to express tumor antigens. Gene therapy has also been used to develop therapeutic products like growth hormones, antibodies, vectors, and recombinant protein. Antibodies for sexually transmitted diseases have been produced, and antibodies created have also helped in diagnosis and therapy of tumors. Hormone production has seen in vitro production of human follicle stimulating hormone through recombinant DNA technology. Recombinant follicle stimulating hormone and luteinizing hormone has helped to enhance ovulation and pregnancy in women. The first example is human insulin. By using the INS gene in producing this hormone, it is inserted into a suitable vector, mainly using E. coli and Saccharomyces cerevisiae. Human insulin is the first licensed drug produced using recombinant DNA technology and was created to cater for the needs of individuals with insulin-dependent diabetes. The next example is human growth hormone. From the GH1 gene, the growth hormone is produced and was done so to help children and adults with growth and development deficiencies, such as Turner's syndrome. Its expression in E. coli allows rapid production in large quantities and is one of the most widely used hormones in supplementation. Unfortunately, it is commonly misused by athletes and bodybuilders to rapidly increase size and strength, and is mainly administered through injections. The last example I would discuss is recombinant factor VIII. Factor VIII is a blood clotting protein and is coded by the F8 gene in humans. It is used for patients with hemophilia A, which is a deficiency in coagulation factor VIII causing prolonged bleeding. Factor VIII activates coagulation factor X, which results in the formation of fibrin-deficient clots. Previous treatment used plasma derivatives, and there were many problems with this approach, where many patients were infected with AIDS through contaminated blood. Currently, however, recombinant factor VIII comes in lyophilized powder form and has overcome these previous issues.
So next we look at the advantages of the technique. The role of recombinant DNA technology in bettering health conditions is exponential. As new vaccines and pharmaceuticals are produced and therapeutic advances are introduced, medicine as we know it is enhanced and improved. Vaccines, for example, are now potentially more economical to obtain, easily distributed, and safer to administer. Vaccines provide invaluable immunity against infectious diseases such as herpes, influenza, hepatitis, and hand, foot, and mouth disease. And as research advances, they may expand further into oncology and other fields. Another advantage is that recombinant proteins play a vital role in combating serious conditions and diseases as they become more prevalent. Insulin, for example, which was once obtained from human and animal tissue, can now be engineered in such a way that it is less expensive to produce and is more readily available. The added benefit is that it has eradicated the possibility of allergic reactions that were once experienced when insulin was obtained from the pancreas of pigs. Insulin is now faster produced and more economical than previous, more conventional methods. Some other advantages include, for example, allograft and dermagraft. These are recombinant products that replace skin for patients of foot ulcers and diabetic ulcers. these are very positive examples of the possibilities of recombination DNA technology in medicine. Also, the creation of transgenic animals have provided medical research with a greater opportunity to study diseases and formulate solutions for combating them. These are genetically engineered mutant animals which are mainly mice. While this is positive for medicine and patients, it does bring with it some ethical considerations, which I will look at over the coming slides. All these points have a positive impact on medicine and look set to continue to advance and improve in the future. Now I'll take a look at the shortcomings of the technique and recombinant protein production. When producing high quality and high quantities of recombinant protein, it has its challenges when expressed in bacterial cells. Barriers to this include protein misfolding and aggregation, resistance in expressing new genes, low solubility and low production, protein toxicity, codon bias, cell stress responses, and instability. Efficiency in the production of functional proteins and choice of vector is paramount to ensure the desired quality of the functional protein. Also, some of the shortcomings are ethical in nature. There are concerns about the manipulation of genetics and the idea of perfecting mankind. This also encompasses the production and use of transgenic animals. Some beg the question, as DNA technology advances, what's in store for our future, and are we in fact playing God? Potential side effects. There have been a number of reported deaths in gene therapy trials, for example the Jesse Gelsinger case in 1999 and 3 other cases since then up to 2007. While gene therapy has vast potential for medicine, it also comes with its own risks. This highlights the fact that research needs to continue to be conducted with complete transparency to ensure safety and efficacy. This is paramount as gene therapy advances. Also, there are concerns for gene doping in sport, which I will touch upon in the following slide. Another ethical consideration is gene doping in sport. WADA, in their 2008 report, defined gene doping as the non-therapeutic use of cells, genes, genetic elements, or modulation of gene expression, having the capacity to enhance athletic performance. As science progresses, athletes now have access to gene doping or hormone therapy in sport, and has become evident particularly in competitive sport and bodybuilding. 3 key substances that are used are erythropoietin, or EPO, which is used for the treatment of anemia in chronic renal disease and is misused in sport to increase endurance as it oxygenates the blood. It is said that there are 3 to 7% of top athletes in endurance sport allegedly using EPO. The Sydney Olympics in 2000 was the first competitive event to employ effective methods to detect recombinant EPO that had been injected. Insulin-like growth factor 1, or IGF-1, and growth hormone, GH, used in medicine to stimulate growth, is also used to increase hypertrophy, or muscle growth, when it is used alongside resistance training. This is popular among bodybuilders as strength and size are enhanced as a result. Hypoxia-inducible factor 1, or HIF1, is associated with the regulation of glucose metabolism in hypoxia, which is a result of low oxygen in the blood. In sport, it is also said to increase endurance in athletes. This practice compromises the ethics of competitive sport, not to mention the possible medical repercussions for those who misuse them. Belinda will now discuss the potential future applications of the technique in the following slide.
Future applications and extensions of recombinant DNA technology and recombinant proteins see the capacity to isolate genes and develop new treatments for other diseases, cancers, and illnesses that can have life-changing ramifications for many patients. Advancements of molecular biology techniques and manipulation of culture environment can predict new strains, vectors, and tags being developed to overcome the limitations of recombinant DNA technology. Those including codon bias, toxicity, protein inactivity, mRNA instability, and lack of post-translational modification. An example of a technique that can be advanced is the CRISPR-Cas system, or the clustered, regularly interspaced short palindromic repeats system. This system is part of the immune system originally discovered in E. coli and is in most archaea and bacteria. In prokaryotes, it relies on short RNAs, which target and silence the unwelcomed DNAs in a specific manner. Applications of this technique are to limit horizon gene transfer and prevent viral invasions. And in using this method, researchers easily make changes including additions, eliminations, or alterations in specific genetic elements of any cell type and organism. Applying this system is still in its infancy, but there are growing trends in its design and application in genome and metabolic engineering. To summarize, with its beginnings in 1969, recombinant proteins are created through recombinant DNA technology and have revolutionized biochemistry and biotechnology, not only in the industries of food, agriculture, and energy, but particularly in the field of medicine and health. While having its advantages, as with many techniques, recombinant DNA technology and recombinant protein production pose some limitations, and therefore do have potential for future advancements in its design and application. That concludes the end of the presentation. Thank you for listening.